For the last 20 years, we've talked about having the complete human genome. It was this landmark achievement, right? It changed science forever. But what if I told you that MAP, our fundamental instruction manual for being human, was never actually finished, that it had gaps, missing pages, right in the spots where some of the most important stories were written?
I mean, just think about that for a second. An instruction manual with entire sections just gone. You could probably still build something, sure, but you'd never understand how it really works, or more importantly, how to fix it when it breaks. For 2 decades, that is exactly the situation we were in with our own DNA. So yeah, let's start right there. The first Human Genome Project was, and I mean this, absolutely monumental. It gave us this reference map that became the foundation for basically all of modern medicine. But that first draft, and even the updates that came after, had thousands of these gaps. It was an incredible map, no doubt, but it wasn't the whole story. And the missing pieces? They were all hiding in the most complicated, trickiest parts of our DNA.
So you have the old map, which we call GRCH38. An amazing piece of work, but it was a composite, kind of stitched together from a bunch of different people, and like we said, full of holes. Now, the breakthrough is this new map, TTTHM13. This one is from a single consistent source, and for the very first time, it is truly gapless. We're talking from the very tip of each chromosome all the way to the other end. And this is so crucial to understand. The missing info wasn't just random static. More than half of all the gaps were crammed into these incredibly dense, super repetitive, and honestly just baffling regions of our genome. These were the parts our technology just couldn't read. It was like trying to navigate a city where an entire neighborhood has street signs that are all identical. You just get lost. So, what was it that made these regions so impossible to map for so long? Well, let's introduce the main character in this whole genetic mystery. Or, maybe the villain of the story if you were one of the scientists trying to solve this thing.
They're called segmental duplications, or SDs for short. Just imagine these long paragraphs of genetic code. We're talking thousands of letters that are copied almost perfectly and then just pasted over and over and over again throughout the genome. Because they're so similar, telling them apart and figuring out the right order was a total nightmare for the old sequencing technology.
The analogy is perfect. It's like trying to piece together a shredded book, where page 50 is nearly identical to page 250 and also page 800. How would you ever know the correct order of the story? The early tech, which read DNA in these tiny little fragments, just got completely confused. It couldn't figure out where each piece of the puzzle was supposed to go.
But that all changed. After years and years of work, this huge collaboration of scientists, the Telomere to Telomere Consortium, or TTT, they finally cracked the code. They built that new map, the one we saw earlier, the TTT-CHM13. And look, don't just take my word for it. The researchers themselves put it best. This wasn't just about plugging holes. It was about getting a fundamentally deeper understanding of our own biology, especially in these really complicated duplicated zones.
The key word here really is gapless, what scientists call a telomere to telomere assembly. A telomere is that little protective cap at the end of a chromosome. So this means they read everything, from one cap all the way down the line to the other cap, with 0 breaks. It's a complete, uninterrupted read of the human instruction manual. Okay, so the map is finally complete. The missing pages have been found. The billion-dollar question is, what was written on them? What kinds of secrets were hiding in that genetic dark matter that we could never, ever read before?
All right, here's the first big reveal, 7%. That is the new estimate for how much of our entire genome is made up of these complex, copy-pasted segmental duplications.
Now, to put that into perspective, we went from thinking about 5.4% of our genome was this duplicated stuff to a full 7%. That jump? That represents about 81 million new base pairs of DNA. That's more than an entire chromosome's worth of new genetic information that was basically invisible to us before now. And this is where it gets even more profound. When the scientists compared this shiny new map to the genomes of 268 diverse people from all over the world, they found it was a dramatically better match. It reflects real human variation in these messy regions way more accurately than the old map ever could. In fact, get this, the new map is 9 times more likely to accurately represent the DNA of diverse people in these complex areas.
This is huge. I mean, absolutely massive. It means we now have a much, much better baseline to study the genetic differences that contribute to our health, to disease, and to everything that makes each of us unique.
And what have we found already? Well, some real game changers. We can now see the full structure of the LPA gene, which is a major risk factor for heart disease. We found this massive expansion of a gene family called TBC1D3, which is linked to the evolution of the human brain. Oh, and we've also identified 182 potential new genes that were just hiding in the gaps this whole time.
Okay, so we've seen the problem, we've met the culprit, and we've seen these incredible new discoveries. But let's just zoom out for a second. Why does having this one complete map truly matter for all of us, day to day? Because this is so much more than just a scientific curiosity, right? It's about laying down a more accurate, a more inclusive, and a much more powerful foundation for the entire future of genetics. It literally changes how we study disease and how we understand our own evolutionary story.
This new map unlocks such a clear path forward. First, we can get way better at identifying genetic risks for diseases. Second, we can dig into the evolutionary journey that made our species what it is today. And third, and this is so important, this is a huge step toward building a pangenome. That's a reference that isn't just based on one sequence, but one that truly represents the incredible, rich tapestry of all human diversity. For 20 years, we were reading an unfinished book. Now, for the first time ever, we have all the pages. And that just leaves us with the most exciting question of all, doesn't it? What other secrets about who we are, where we came from, and where we're going are just waiting for us, hiding in the story that we can finally read in its entirety?